In 1968, a film called Planet of the Apes asked a question that had been sitting in the back of human culture for a long time. If apes are so close to us, if they use tools, form social groups, solve problems, and communicate, what exactly is stopping them from taking over? The film treated it as science fiction. The genome treats it as a genetics question, and genetics has an answer. Humans and chimpanzees share 98.7% of their DNA. This number gets quoted in every documentary, every biology class, every conversation about human uniqueness. The implication is that we are almost the same, 1.3% away, a small step. That framing is wrong, and it is wrong in an interesting direction. The gap between a chimpanzee and a human is not small. It just looks small when you measure it the way most people measure it. When you measure it the right way, by asking what the 1.3% actually contains, what it does, and how long it took to accumulate, the gap is not a step. It is a staircase, and we are still climbing it. The case is not about what chimps are missing, it is about what it would actually take to close that gap. And the answer from the DNA is more specific, more surprising, and more final than the question expects. First, the chimps deserve credit for what they already have. Chimpanzees use tools. Wild populations in West Africa use stone hammers and anvils to crack open nuts, a technique that requires understanding the relationship between force, angle, and target, and which takes juveniles years to learn from adults. Different chimp populations have different tool traditions that do not spread between groups. Cultural transmission of learned behavior, not just instinct. Chimpanzees communicate. They have dozens of distinct vocalizations tied to specific situations. Predator alarm calls that distinguish aerial from ground threats, food calls, social signals. They gesture, they deceive. In captivity, chimpanzees have learned to use hundreds of symbols to communicate requests and observations. They understand referential communication, that a symbol or a gesture points to something in the world. Chimpanzees plan. Experiments have shown that they can select and carry tools to a location where they will need them later, demonstrating a mental model of a future situation they are not currently in. By every behavioral measure we applied to human ancestors to call them cognitive predecessors, chimpanzees qualify. And yet something stopped. The question Planet of the Apes asks is why, and whether it could still happen. The genome has a specific answer. In 2001, researchers studying a British family with a severe and unusual speech disorder traced the condition to a mutation in a gene called FOXP2. Affected family members had profound difficulty producing and understanding grammatically complex language. Not a motor problem, not an intellectual disability, but a specific disruption to the neural wiring of language itself. When researchers sequenced FOXP2 across primates, they found something unexpected. The human version of FOXP2 differs from the chimpanzee version at two specific positions. Just two. Those two changes are present in every human being on Earth, and in no other primate. They appear to have been under intense positive selection within the last few 100,000 years, concentrated in the period when anatomically modern humans were emerging and behavioral complexity was accelerating. Those two amino acid changes do not add language to the brain. What they do is change which genes FOXP2 switches on and off during brain development. FOXP2 is a transcription factor, a master switch that regulates hundreds of other genes. The two changed positions in the human version alter the entire downstream program, the development of neural circuits involved in fine motor control of the face and mouth, auditory feedback processing, and the learning of complex sequential movements. Those circuits are, together, what makes speech physically possible. Chimps have FOXP2. They do not have the two human-specific changes. Their version of the gene runs a slightly different developmental program. Not a broken program, a different one. The chimp brain and vocal tract are wired for what they need to communicate in a chimp social environment. Wiring them for human language would require not just those two amino acid changes, but the entire cascade of developmental consequences those changes set in motion over hundreds of thousands of years. And that is only one gene. There are 201 more where it gets complicated. When researchers compared the human and chimpanzee genomes looking for sequences that changed far more rapidly in the human lineage than in any other mammal, they found 202 such regions. These are not genes, they are regulatory sequences, enhancers and switches that control when, where, and how intensely nearby genes are expressed during development. Most of them were nearly unchanged across all mammals for tens of millions of years. Then, in the human lineage specifically, they changed. The most studied of these is called HACNS1. It is active in developing human limb buds and in specific brain regions during embryonic development. When researchers replaced the human version with the chimpanzee version in developing mouse embryos, the gene expression pattern in the limb changed measurably. The human version produces a different developmental program in the hand, not a better hand in an abstract sense. A different hand. One with a specific precision grip configuration that our lineage needed, and the ancestral primate hand was not delivering. A third piece of evidence involves a gene called sRGAP2. In the human lineage, this gene was duplicated approximately 2.4 million years ago. The duplicate copy interferes with the original. The original sRGAP2 protein normally puts a speed limit on how quickly neural connections mature in the developing brain. The duplicate jams that speed limit. Human neurons develop more slowly and end up with more connections per cell than chimpanzee neurons. This matters for the Planet of the Apes question more than any other finding. That slower brain development is not a deficiency. It is the source of human learning capacity. A brain that is slow to mature stays plastic for longer. A child who spends years in a state of neural flexibility can absorb language, culture, technology, and social complexity in a way an animal whose brain matures quickly cannot. The extended human childhood, which makes us helpless for years and dependent on our parents for a decade, is the price of that plasticity. It is also the mechanism behind most of what makes human civilization possible. The sRGAP2 duplication happened approximately 2.4 million years ago. The first stone tools in the archaeological record appear at approximately the same time. Whether one caused the other cannot be proved from genetics alone. But the question is worth sitting with. Here is what Planet of the Apes gets wrong, and it is a specific genomic wrongness. The film assumes the gap is small enough that a push in the right direction, a virus, a drug, a brief period of forced evolution, could close it quickly. The genome says the gap is not one thing. It is a web of interdependent changes across 202 regulatory regions, at least 2 gene duplications, and dozens of gene losses, all of which accumulated over 6 to 7 million years in response to specific environmental pressures that are not currently operating on chimpanzees. The FOXP2 changes happened in the context of social groups that placed enormous fitness value on precise communication. The HACNS1 changes happened in the context of selection for the kind of fine manual dexterity that tool use rewards. The sRGAP2 duplication happened in the context of an environment that rewarded an extended learning period. These were not random changes. They were selected because they helped specific ancestors survive specific problems. Chimpanzees are not in those environments. They are in forests, in the specific ecological niche they have occupied for millions of years, accumulating their own variants in response to their own pressures. They are evolving. Some chimpanzee populations already show the early stages of stone tool culture that have persisted for thousands of years. The question is not whether chimpanzees could ever develop more complex cognition. The question is whether the specific combination of pressures that produced human cognition could ever produce the same result twice. The genome says, not on our current timeline, and not in the direction the films imagine. But the more honest answer is that we cannot predict what 6 million years of the right pressures would do to a chimpanzee lineage. We know what they did to ours. The 202 human accelerated regions have been catalogued but not fully understood. Each one controls a potentially different developmental program. Knowing that HACNS1 changes limb development is the beginning of the story, not the end. The full downstream effect of each regulatory change on the anatomy of the organism that develops under its influence is still being mapped. There is also the question of what happened to the chimpanzee lineage during the same 6 to 7 million years. The chimpanzee genome is not the ancestral genome. It has accumulated its own derived changes, its own regulatory shifts, its own duplications and losses. The common ancestor we share with chimpanzees was not a chimpanzee and was not a human. Reconstructing what it looked like genetically, and therefore what changes occurred in each lineage independently, requires comparison across multiple primate species and is still ongoing. The disease divergence question is also live in both directions. Chimpanzees infected with HIV do not progress to AIDS. Chimpanzees do not develop Alzheimer's disease in the human pattern. The genetic differences that account for these divergences are not fully understood and have direct medical relevance. The same 1.3% that separates our cognition also separates our disease vulnerabilities. Some of what makes us human makes us specifically fragile in ways chimps are not. Planet of the Apes was science fiction. The genome makes it more complicated than that. Chimps are not almost human. They are not stuck one step below us on a ladder we both share. They are on their own trajectory, accumulating their own variants, adapting to their own world. The 1.3% that separates us is not a small gap in a large similarity. It is a record of 6 to 7 million years of divergence concentrated exactly in the systems that matter most: language circuitry, hand development, brain growth rate, the extended childhood that makes culture transmissible across generations. 2 amino acid changes in FOXP2, a regulatory switch in HACNS1 that wired a different hand, a gene duplication in SRGAP2 that slowed the human brain down and left it plastic for years. 202 regulatory regions that changed faster in our lineage than in any mammal that came before us. Dozens of gene losses that stripped away programs our ancestors no longer needed. None of these happened because a virus rewrote the chimp genome overnight. They happened one selected variant at a time, across millions of years, under pressures that shaped what those changes were good for. The staircase between a chimpanzee and a human was built one step at a time, and each step took thousands of generations. Chimpanzees are not going to take over. Not because they are incapable of evolving, but because they are already evolving toward being better at being chimpanzees, which is its own kind of remarkable. And the 1.3% that sits between them and us is not a locked door. It is a record of a very long journey that we are still on. The case is not about what separates us. It is about what it took to build the difference. And the answer is everything. 7 million years of everything.